So today, I'm going to talk about really the details of about the single cell ATAC-Seq data analysis. So you probably have seen this type of figures many, many times, right? This is just a type of the visualization for single cell analysis, either single cell RNA-seq or ATAC-Seq. And you probably may have wondered what it takes to produce this type of figure from raw sequencing data. So the goal of today's primer is to really help you fill in these gaps, okay? Fill in these gaps so that you better understand next time you read a paper or listen to the talk about the single cell ATAC-Seq. Okay, so first of all, what is ATAC-Seq and how it works? So ATAC-Seq is an assay for transposase accessible chromatin with high throughput sequencing, originally developed by Jason Brenner-Stroh in 2013 in Greenleaf Lab. And it is now one of the most popular methods to detect the open chromatin region in the genome. So the ATAC-Seq experiment starts by treating the intact nuclei with the TM5 transposase, which are preloaded with the sequencing adapters, shown here, red and green string. Then once these transposase binds to the DNA, then this enzyme actually cuts the DNA and then adds the sequencing adapters at the end of the fragmented DNA. So it's essentially the cut and paste at the same time. And this process is called tegmentation. And importantly, this reaction is mostly restricted to the open chromatin regions shown here, because the transposase cannot go easily into this closed chromatin. So as a result, most of the DNA fragment generated by this process are from the accessible chromatin. So these fragments are then collected and amplified and sequenced, and then finally aligned to the reference genome to figure out which regions are really open. So there are a couple of things to note from this figure. First, not all fragments created by TM5 transposase are actually amplified and then sequenced. It only amplified when you have the right combination of the sequencing adapters, okay? One end has to be green, the other end has to be red. Secondly, the initial material is actually destroyed, meaning once it's tegmented, then the portion of the DNA that they contribute that fragment are gone. So you no longer be able to create the fragment overlapping that part. So those are the two things to keep in mind. So here's an example. This is the IGV genome browser showing the sequenced reads from the bulk ATAC-Seq data mapped to the human genome. Each dumbbell shape here, red, dumbbell shape, the red or blue are paired end reads and aligned to this particular genome. And also here, the top track shows the density of the read mapping at the particular position. So I think the more accessible region should have more read mapped to them, resulting in higher density. So this leads to an important concept in ATAC-Seq, which is open chromatin peak, because the open chromatin region actually comes as a form of peak. As the name suggests, these open chromatin peaks are regions that have significantly higher read density compared to the other regions. So finding these peaks is one of the primary goals of ATAC-Seq data analysis, okay? All right, and then these are the regions that are identified by peak calling algorithm, which very nicely aligned to this open chromatin peaks. All right. So far I have described, yes, please. Yep.
how should we understand the breadth of the peak, the border of the peak? So, um, yeah, what's the significance of that when I'm looking at these things, trying to understand? So I understand there's a bimodal thing, different reads are attaching, but many times I have seen the length change. What is that telling us, and how to understand it from a point of view, what's biologically happening, or it's an artifact of something? How do we distinguish at that time?
So I think it's more about the biology. So if the regions are broadly open, then the correspondingly the width of the peak should also be broader. So I think the peak calling algorithm actually try to also capture those aspects when they call the peaks and then identify peaks.
So taking max to an example,
Yep.
which I think is the most common one.
Yes. I use that all the time.
Right. So do you use like in that case like default settings, or like I'm just thinking from where should I be concerned? That's what my major question is in here, because when you mention max 2 now, I'm curious to know, because I have played with that and I'm never ever sure when to know or how to know actually something is messed up.
Yes. So Max2 is, I mean, as I said, and as you said, it's one of the most popular tools to call the peaks. Even if it was originally developed for the analysis of the different types of the, genomic data, ChIP-seq, but it's actually works really well for the analysis of the ATAC-Seq. But you have to use the specific combination of the parameters in order to make it work on the ATAC-Seq. And also, for the ATAC-Seq analysis, Max2 is actually equipped with the estimation of the read shift mapping to the negative strand and then plus strand that are specific to the ChIP-seq. That doesn't apply to the ATAC-Seq. So you really have to use the specific set of parameters in order to use Max for the ATAC-Seq. Now, there are also parameters. When you do that, you really have to specify a shift size and then fragment size and et cetera, but it has been heavily discussed by other pipeline. And then my suggestion is that you should stick to the just popular parameter sets that are used by other people. Okay, great question. All right, so far I have described the bulk ATAC-Seq, right? Then what about the single cell ATAC-Seq? Actually, the single cell ATAC-Seq is conceptually the same as bulk ATAC-Seq. The only major difference between the single cell ATAC-Seq and a bulk ATAC-Seq is that in single cell, a unique cell barcode is assigned to each fragment. That's the only difference. And single cell ATAC-Seq is also often called single nucleus ATAC-Seq because genomic DNA is only present in the nuclei. The experiments are typically performed on the extracted nuclei, so rather than the dissociated cells. So that's why it's called a single nuclear ATAC-Seq. But in this talk, I'll not distinguish between the two. Okay. Okay. So experimentally, then how we attach or assign the cell-specific DNA barcode to the ATAC-Seq fragment. So there are two basic approaches. The first is the combinatorial indexing-based technique, and then second one is the microfluidics-based approaches. So let's look at the combinatorial index-based approach first. So the basic idea is that we will assign a unique combination of the barcodes to each cell by repeatedly pulling and splitting the cells after adding the barcodes. Okay, so pulling. So in this toy example, yeah, well, I only have like 16 cells, right? In 4 wells, equally divided. So in this situation, we can uniquely label these 16 cells with 4 different barcodes, okay? With the 2 rounds of the barcode addition. So here you add a unique barcode to each well to these 4 cells. And then pull and split, and assuming that they are randomly distributed. And then once you add the second barcode, then combination of the first and second barcode uniquely determine each cell. So that's the basic idea. The biggest advantage of this method is that it's highly scalable and then also very cost effective. However, it's potentially less sensitive because of the multiple rounds and the complex steps of the library preparation. So those are the two major pros and cons of this method, this approach. The another approach is based on the microfluidic techniques. This method can be further divided into the two different platforms. The first method is a plate-based approach where microfluidic chips is designed to directly capture single individual cell and place each of them into a microwell, here, shown here, microwell on a plate, and then perform a ATAC-Seq on each microwell separately. So this can generate really high quality reads per cell, but the throughput is really limited because the maximum number of cells that you can evaluate per experiment is the number of wells in a plate, so which is typically 96 or in a bigger scale, 384. So the second approach is a droplet-based approach where each droplet is designed to capture single cell and then also contains the unique barcodes in it. So as you can see here, the beads containing the unique barcode flowing through this channel, and then cells flowing through this channel, and then they made it, statistically just once in a while. It just combined together, and then single cell is supposed to capture to one bead at a time. So this provides a very good sensitivity. Well, I shouldn't say very good. That's a reasonably good sensitivity, and also reasonably high throughput. So this 10x genomics platform has become one of the most popular assays these days. All right. So I will now switch gears to discuss each of the processing steps, as I promised you, from FASTQ files all the way to the cell cluster in much more detail. So in this workflow, I try to group these common processing steps into just 5 main tasks, as shown here. But then later, I realized that these can be combined together. Anyway, namely, trimming and alignment, and then read QC filtering, and then cell QC filtering, and then after that, dimensionality reduction, and finally, clustering and visualization. Okay, let's take a closer look at each task. So the first and second task are actually quite straightforward and should be familiar to many of you guys because they are common to the high throughput sequencing data analysis, such as whole genome sequencing and RNA-seq. The first step is adapter trimming. The adapter sequences for ATAC-Seq should be trimmed because many of the fragments generated by ATAC-Seq is actually shorter than read length these days. Okay, the shortest possible fragment size is 35 to 37 base pairs, and the typical read length is 50 base pair. So even for the and then actually more than 20% of the reads are actually shorter than 50. So which means almost more than 20% of the reads are expected to contain this adapter part of the adapter sequence. So they should be removed before mapping to the reference genome. So once these adapter sequences are trimmed from your read, you can then use any standard short read aligners to map these reads to the reference genome. So I'm not going to get into the detail of this part. And also, and then once all the reads are mapped to the genome, then you can then perform the standard read QC to really filter out the low quality and less informative reads. So typically, low mapping quality reads or the duplicated reads, mitochondrial reads, and the chimeric reads are, et cetera, are typically excluded from the analysis. Okay, so these are all just common to most NGS analysis. Okay, so after the read QC, we then convert the BAM files to a fragment file, which is essentially a list of the fragment generated by the ATAC-Seq experiment across all cells. So it has 5 columns as shown here: chromosome, start and end, and then also barcode, which corresponds to the specific cell, and then a number of fragments that match to this particular coordinate. So one thing I like to note here is that the start and end coordinates may not exactly match to the read coordinate in the BAM file, especially if this fragment file is generated by the 10x cell range or ATAC-Seq analysis pipeline. I think the reason is actually very biologically driven. So it is actually well known that TM5 transposase adds this additional 9 base pairs during the process of adding these adapters to the fragmented DNA. So if you really want to identify the central position that are detected by this TM5 transposase, you have to account for that. And what is typically done is that the start position of the fragment, which is on the left side, or the start, is shifted toward inside of the fragment by 4 base pairs. And then the end position is again shifted toward left side by 5 base pairs, okay? So that is already implemented in the 10x cell range ATAC-Seq pipeline. As a result, the fragment file generated by that pipeline is already adjusted. So that's something you have to remember. Okay. So now that we have high quality fragments adjusted by these biological process across all cells, and we now are ready to look at the cells, okay? So there are many things to consider for cell QC, but there are two most important metrics. First is the number of fragment in a cell, and then second is the proportion of the reads that are mapped to the known or regulatory elements, okay? So there are also several different ways of measuring this, but transcription start site enrichment score is one of the most widely used metric to evaluate the quality of the cell. So how does it work? So in Surat, and then other tools actually use very similar formula, but the TSS enrichment score is basically is the fold change calculation, okay? You first count the number of fragments that fall within the signal region, which is typically defined as 1,000 base pair centered in the transcription start site, okay? And then this number of fragments mapped to the signal region is normalized by the number of reads falling in the flanking region, which is actually 2 kb, 1 kb away from the transcription start site, and then 100 base pair region. So the number of fragments falling in this divided by the sum of the number of the fragment falling in these 2 flanking regions. So that's your transcription enrichment score calculated by the Surat. I checked the code, so this is correct. All right. So the popular visualization strategy for these 2 metrics is to look at them in a scatter plot, as shown here. X-axis is the number of fragments actually in peaks. So you can either use all fragments that are found in a cell, or you can additionally filter fragment based on the overlaps with the open chromatin peaks. So in this case, in this figure, we used a number of fragments that are found within peaks. And the y-axis is the transcription start site enrichment score that I just described in the previous slides. And each dot represents a cell, and the brighter color represents the multiple overlapping cells, higher density. And the red lines are essentially just the percentile. This is the 5th percentile, 10th percentile, the 90th percentile, and 95th percentile. Obviously, we don't want cells with a very low fragment count. So all the cells that are on the left side in this figure are likely to filter out. Also, we don't also want to cells that have extremely high feed count because they are potentially doublet, which I'm going to talk about separately. And again, the TSS enrichment also follow the same trend, except that we also keep the cells with the high transcription start TSS enrichment score. All right. So we can also evaluate the other metric with just simple violin plots. Here is the distribution of the 5 different metrics that are commonly considered. I directly grabbed this figure from this Surad vignette for PBMC dataset. Okay, so I mean, I just want you to look at how they are, distributed. This widely vary. The other thing I like to note here is that it is almost impossible to find a single cutoff that applies to the multiple data set, meaning the distribution of these metrics are actually different for different data sets. Okay? Even if you try to combine them, it is highly recommended that these metrics should be evaluated separately for each data set, and then cutoff should be determined separately for each data set. Okay? All right. So as I briefly mentioned before, another potential significant artifact is a doublet, which is defined as a droplet containing 2 cells. Okay? But computationally speaking, it is essentially a cell barcode associated with fragment originating from 2 cells. Okay? Okay. So the other thing is the other important concept is the heteropic doublet, which is the doublet that are joined by 2 different cell types. So this one is actually can lead to the wrong cell types and then just complicate all the downstream analysis. So this has to be excluded from the analysis. But it's hard to detect such doublets because of the sparse nature of the data and others, and then other technical difficulties. So in order to detect these doublets, there are generally 2 main ideas have been tossed around. The first is actually originally developed for the analysis of the single-cell RNA-seq data. The idea is that it generates artificial doublets by combining the 2 randomly sampled cells and then using them as a basis to find the real doublet in your data set. So you just to repeat many, many times to figure out which cells are potentially doublets. So there are many tools that work really well using these ideas. The second idea is more interesting. This is specific to the ATAC-Seq, and it actually leverages the fact that the diploid cells can only generate up to 2 overlapping fragments. Remember that ATAC-Seq is the once it's generated by the ATAC-Seq, it's gone. The material is gone. It cannot be generated one more time, right? So using this idea, you can actually ask how many reads are actually overlapping per cell for any particular regions. And if you find more than 2 overlapping reads many, many times in a cell, then it is likely that those barcodes are probably doublets.
May I ask a question?
Please.
Do you first mask part of the genome where it has low mappability, where you would just get multi-mapping just because of the genome reference, not because there are actually two cells in that droplet?
Oh, so we only use the high quality reads for this doublet removal analysis. So yes, because of that issue, the reads with low mapping quality actually complicated this step. So we have to remove that. I think that works actually better than including them in these steps. All right.
So you mentioned about chimeric read. So is there a case where if you see after looking the alignment and you see high chimeric rate, could that be an indication of that you have doublet, or that's an indication of something else is happening where you are seeing high mapping to chimeric, two different regions?
We never looked at it in that way, but I suspect that the chimeric reads is unlikely produced by doublets because it's the same genome essentially, right? So, okay, that's an interesting question. Thank you. All right, so these we typically use these tools together in our pipeline. So, because each method actually captures a quite different proportion of the doublets. So we found that it works best when we combine these two methods together. Okay, now we have high quality fragments and also high quality cells. Okay, now it's time to build another primary data set, which is feature cell count matrix. So for those who have some experience with the single cell RNA-seq, you probably know the gene cell count matrix, right? So you have the genes on the rows, and then cells on the columns, and then you basically count how many genes transcripts are appeared in each cell. So we want to do the very similar things with a single cell ATAC-Seq data. The problem is there is no naturally well-defined feature sets for the single cell ATAC-Seq. There are many different types of the features that can be used to define this feature set count matrix, but there are two major classes. One is the region driven from the data by identifying open chromatin peaks first from the aggregated reads, and then use that region to really as a feature. Secondly, you can divide the entire genome into the fixed length window and then use that as a feature. So there are pros and cons for these two methods, but essentially those are the two major type of approaches for building the feature cell count matrix. Okay, either way, it will eventually create the feature cell count. And then as shown here, the cell is the column is the cells and the row is the peaks. And then each cell is containing the number of fragments that fall in that peaks for the particular cells. But number of features, speaking about the number of features, they are very, very large. Even for the number of peaks, you typically find more than 100,000 peaks per sample. And beans are even worse. There are 60 million beans if you use 500 window size bean for the entire genome. But luckily, many of these features in this count matrix are correlated. And then it can be, and then dimensionality reduction techniques can be applied to really reduce and then compress the data. That's what I'm going to talk about. So the goal of the dimensionality reduction step is to really transform the original feature cell count matrix that I described in the previous slides into a new count matrix with a manageable number of features. Usually K equals 32, 50, or the 100, okay? So in this step, but also you have to preserve the information that are encoded in the original feature set count matrix as much as possible. So in this step, normalization, the transformation of the matrix, and even the feature selection can be applied in conjunction with the actual dimensional reduction methods. And also, in case you have multiple samples, as I just briefly mentioned in the previous slides, you would expect batch effects, right? You cannot avoid the batch effect. And this is the step where you should perform the batch correction, okay? So for the particular dimensionality reduction method, there are many methods as well, but I will focus on one specific method, but the idea is all the same. But the particular method that I'm going to describe today is the latent semantic indexing, or the LSI. It has the three major steps. First, it's a simple binarization of the count matrix, and then filtering out low information row, and then also optionally depth normalization is applied. Secondly, the TFIDF transformation is applied to this count feature matrix. And then finally, the singular value decomposition is applied to the matrix. It was originally introduced by this paper in 2015 and had been implemented in the many popular software tools. So TF-IDF is originally developed actually in the field of the natural language processing to really evaluate how relevant a word is to a document that you're looking at. So all the terms are really relevant to the language processing. TF is an acronym for the term frequency. IDF is acronym for the inverse document frequency. But in the single-cell ATAC-Seq setting, peak or bin, the features, is equivalent to the word, okay? And then cells are document, okay? That's how you translate this term. But that's still, it's hard to grasp the idea. So I made a very simple toy examples. So here's a toy matrix of the already binarized count, binarized, with the 4 peaks, okay? And then the 5 cells. So using these toy examples, I'm going to show you how to calculate TF-IDF. So first is TF. It is essentially per cell normalization, meaning the peaks in each cell. So in this particular cell, there are 3 peaks, okay? It's normalized by the total number of peaks. So it's a per column normalization, okay? So IDF, although it sounds very fancy, but it's actually per row normalization. So for each peak, you first count how many cells actually containing these peaks, and then that's the term that is for the normalization. So that's shown on here. And then TF-IDF is essentially just a combination of these two. It's a cell-wise calculation. So intuitively, it's essentially provide more ways to rare peaks. And then you will get even more weights if the peaks are present in rare cell type, something like that. So that's the idea. So once TF-IDF transformation is done, the next step is the matrix factorization using singular value decomposition method. So the PCA, you probably know PCA, right? The PCA is a special case of the SVD. It's essentially decomposing a input into the multiplication of the 3 matrices. And then this is U and V are orthogonal matrix, and then the sigma is a diagonal matrix. So, but then this doesn't really do anything about the feature selection or the dimensional reduction. What we actually apply is truncated SVD. So instead of using full matrix, we only select the features that have a certain, that capture certain proportion of the variance. So in this situation, instead of capturing full matrix, we only care about the first k number of features to approximate the original matrix. So sometimes it's called embedding features and cells into these k features, but to me, I don't really like the embedding term when it comes to describing this. Anyway, in the end, the k, by the way, k is the user-defined parameters, and then typically used to 50, and sometimes you can go more, like 100, or you can go fewer, 30, depending on how many cells you have and how complex the data are. But in the end, you will have the n by k matrix. So for each cell is represented by the k number of features. Okay, so you're actually going to use this matrix for as a reduced and transformed feature cell count matrix. All right. Okay. So we're almost done. We're almost done. The clustering. Now that we have really manageable feature cell count matrix, we are now ready to perform I clustering. Okay. But this is, you can think of a high-dimensional space, and cells are actually scattered in that high-dimensional space, okay? And then in order to perform this clustering, it actually works much better if you have a connected graph in this high-dimensional space. So you can think of this hypothetical graph. Cells are nodes are cells, actually. And then you try to connect between cells if cells are close to each other in this high-dimensional space, okay? So k nearest neighbors are essentially, it's very simple. It's k nearest neighbor is essentially just connect, for each cells, you connect the k neighbors that are closest to that cell in that space. Shared k nearest neighbors, it has one more criteria, which is essentially instead of connecting all the k neighbors, you only connect the two cells if these two cells also share the neighbors in this k nearest neighbor space. So here, again, another toy example. Left is, I'm going to build a k nearest neighbor graph on the left, and then shared k nearest neighbor graph will be built on the right side with k equal 3. And for shared nearest neighbor, I'm going to, as soon as we have one neighbor, I'm going to call it, okay, this is shared nearest neighbor. So let's look at this cell first. These are the 3 nearest neighbors, okay? Now let's look at this cell. And then these are the 3 nearest neighbors, okay? Since these 2 cells share the 2 neighbors, more than 1, right? These 2 cells will be connected in shared nearest neighbor graph, okay? Now let's look at this cell. The 3 nearest neighbors will be this, but these 2 cells don't share any neighbors, right? In this nearest graph. So even if they are considered, but it ended up, pruned out of this shared nearest neighbor graph. So we repeat this process, and this is what you get, all right? So in the end, shared nearest neighbor will give you a much more compact and a highly well-connected community compared to the k nearest neighbor. So this, the shared nearest neighbor graph is typically used and then implemented in many ATAC-Seq. And it's actually not specific to the single-cell ATAC-Seq. It's implemented for the single-cell RNA-seq data as well. Okay, so now we have a graph. What's next? Just perform the clustering algorithm. Hierarchical clustering and then k-means clustering is the two most widely used method. For the hierarchical clustering, Levine algorithm and Leiden algorithms are the most popular clustering algorithm. And it's a greedy algorithm that merge the nodes together if they are well connected. And Leiden algorithm is just a modified version of the Levine algorithm, which almost guarantee that identified clusters are well connected to each other. So that's the only difference. And then these are the two references. Okay. Well, we haven't talked about the visualization, right? Visualization is really tricky, tricky method, a really tricky step. It is known that it is mathematically impossible to preserve the distance relationship between cells, okay? When projecting the data in high-dimensional space into the two-dimensional space. It's not possible. You will end up breaking some of them. But there are useful methods that preserve this structure as much as possible. Those are the t-SNEs. Those are the t-SNEs and UMAP. And then those are the two most popular methods that are currently used for visualization. The input data is the reduced and then transformed counter matrix that I described that goes into this either t-SNE or the UMAP algorithm that will spit out the coordinate two-dimensional or the three-dimensional space, okay? One thing I like to note here is that clustering algorithm is nothing to do with the visualization, okay? They use the same input, but it's a completely two different algorithms applied to the clustering and then also visualization. So that's why oftentimes when you overlay the clustering labels to this t-SNE or UMAP visualization, some of them are not really matched to each other. That's why. So those are the important to remember. Okay, now I hope you have a better understanding of these computational steps of processing the single-cell ATAC-Seq data from FASTQ files to produce the clustering. But you know that this is the beginning of the single-cell ATAC-Seq data analysis, right? There are all kinds of the downstream analysis that you can play with using this clustering. So you have to annotate the cells. You can rely on the marker gene activity, or you can also rely on the single-cell RNA-seq data to transfer the label to the ATAC-Seq. You can also do the differential accessibility analysis across the different cells. You can also apply the different clustering or the clustering algorithm so that you can find just the pseudo-time or the trajectory of the cell differentiation or cells proliferation or whatever. And you can also look at the correlation between the chromatin accessibility across the cells to figure out which peaks are actually correlated. Okay. That helps you to build some potential gene regulatory network. You can also look at the regions that are identified by this single cell to figure out which transcription factor binding sites are enriched. And finally, you can integrate this data with the single-cell RNA-seq data to really build a gene regulatory network. And these days, you can even generate a single-cell ATAC-Seq and RNA-seq from the same nuclei called a multiome. So this is my last slide. Here is the summary of some of the most popular tools used to analyze the single-cell ATAC-Seq data. I mean, this is by no means the exhaustive list of the tools, but to just highlight a few, where is it? The SIGNAC is actually the ATAC-Seq version of the Seurat, and it actually borrows a lot of functionality from the Seurat package as well. RCHAR actually has excellent doublet detection algorithm implemented. Where is it? SnapAttack is implemented in Python. It's extremely fast, and then we actually use it. Cis-topic is known to produce really robust clustering result. So this one is also used quite open by many people. Okay, with that, I'll stop my talk here and I'll get any additional question if you have.
All right. Well, thank you so much for that really great introduction to single nucleus ATAC-Seq.
Thank you.